Cell organelles. Every cell has a membrane around it to keep its own content separate from the external environment. Multicellular organisms like plants and animals need a lot of chemical activities to support their complicated structure and function. To keep these activities of different kinds separate from each other, these cells use membrane bound little structures called cell organelles. Some important examples of cell organelles which we will discuss now are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles. They are important because they carry out some very crucial functions in cells. Let's discuss them one by one. Endoplasmic reticulum. The endoplasmic reticulum is a large network of membrane bound tubes and sheets. It looks like long tubules or round or oblong bags called vesicles. The endoplasmic membrane is similar in structure to the plasma membrane. It extends throughout the cytoplasm. There are two types of endoplasmic reticulum: rough endoplasmic reticulum RER and smooth endoplasmic reticulum SER. What is the function of endoplasmic reticulum? It serves as channels for the transport of materials, especially proteins, between various regions of the cytoplasm or between the cytoplasm and the nucleus. It also functions as a cytoplasmic framework, providing a surface for biochemical activities. Rough endoplasmic reticulum. Rough endoplasmic reticulum are arranged into flattened sacs, as you can see in this diagram. They are rough because ribosomes are attached on their surface, thereby giving it a rough appearance. Ribosomes are the sites of protein synthesis. Cells that specialize in secreting proteins have lots of rough endoplasmic reticulum. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum is a series of interconnected tubules. There are no ribosomes attached on their surface. A smooth endoplasmic reticulum helps in the manufacture of fat molecules or lipids. In liver cells of vertebrates, smooth endoplasmic reticulum detoxifies many poisons and drugs. Membrane biogenesis. We know that endoplasmic reticulum helps in the manufacture of proteins and lipids. Some of these proteins and lipids help in building the cell membrane. This process is known as membrane biogenesis. Thus, membrane biogenesis is the process of formation of cell membrane from proteins and lipids, while some other proteins and lipids function as enzymes and hormones. Next is Golgi apparatus. The Golgi apparatus was first described by Camillo Golgi. It consists of a system of membrane bound vesicles which are arranged approximately parallel to each other in stacks called cisterns. These membranes often have connections with the membranes of endoplasmic reticulum and therefore constitute another portion of a complex cellular membrane system. The material synthesized near the endoplasmic reticulum is packaged and dispatched or transported to various targets inside and outside the cell through the Golgi apparatus. Its functions include storage, modification, and packaging of products in vesicles. The Golgi apparatus is also involved in the formation of the lysosomes. What are lysosomes? Lysosomes are a kind of waste disposal system of the cell. Lysosomes help to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Foreign materials entering the cell, such as bacteria or food, as well as old organelles, end up in the lysosomes, which break them up into small pieces. Lysosomes are able to do this because they contain powerful digestive enzymes which are capable of breaking down all organic material. During the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosomes may break up and enzymes digest their own cell. Thus, they are known as suicide bags of a cell. Structurally, lysosomes are membrane bound sacs which are filled with digestive enzymes, and these enzymes are made by rough endoplasmic reticulum. Next cell organelle is mitochondria. Mitochondria are known as the powerhouse of the cell. The energy which is required for various chemical activities is released by the mitochondria in the form of ATP molecules. ATP stands for adenosine triphosphate molecules. ATP is known as the energy currency of the cell. Mitochondria have two membranes instead of just one. The outer membrane is very porous while the inner membrane is deeply folded. These folds create a large surface area for ATP generating chemical reactions. Mitochondria are strange organelles in the sense that they have their own DNA and ribosomes. Therefore, mitochondria are able to make some of their own proteins. Plastids. Plastids are present only in plant cells. They are divided into two categories, chromoplasts and leucoplasts. Chromoplasts are the colored plastids, whereas leucoplasts are the colorless plastids. Plastids containing green pigment chlorophyll are called chloroplasts. Leucoplasts store materials such as starch, oils, and protein granules. If we talk about the internal organization of plastids, plastids also contain membrane-bound layers embedded in a material called stroma. Plastids has a similar structure like the mitochondria. They also have their own DNA and ribosomes. Vacuoles. Vacuoles are the storage sacs for solid or liquid contents. Vacuoles are small sized in animal cells, whereas plant cells have large size vacuoles. The central vacuole of some plant cells may occupy 50 to 90% of the cell volume. In plant cells, vacuoles are full of cell sap and provide rigidity and turgidity to the cell. Many substances of importance in the life of plant cell are stored in vacuoles. These include amino acids, sugars, various organic acids, and some proteins. In single-celled organisms like amoeba, the food vacuole contains the food items that the amoeba has consumed. In some unicellular organisms, specialized vacuoles also play important roles in expelling excess water and some waste from the cell. So, in the last, the cross-section of plant cell and animal cell is given. From these diagrams, you can easily make out the comparison between the two. Plant cells have cell wall, whereas animal cells have only cell membrane. The vacuoles in case of plant cells is of large size, whereas animal cells have small sized vacuoles. You have to make out the differences and similarities between the two cells and write it on the notebook.